Okay, before we get started today, I want to talk about our schedule for our third exam. Okay, our third exam is scheduled for the 15th, which is next Wednesday. Okay, that isn't when Capstone Day, is it? Capstone Day is the following week, right? Okay, so there's no conflict there. I did have a request from a couple of students on Wednesday to move the test to Friday. I don't think that would be a good idea because baseball's going to be gone. They have an away game that weekend. They have a doubleheader, tripleheader. Softball's going to be gone that weekend. Track's going to be gone that weekend. And I don't want to be giving 25 makeups, okay? I don't know about, let's see, what's the other team? Golf. I don't know about golf.
I don't even know.
But there's only, I think we only have one person on the golf team here, so. Now, we are a day behind because we had a snow day. What was it, right after spring break, I think? So we missed a day of class then. That wasn't something I had planned on. So, I think we are, I do need to postpone the test, but we can't do it on Friday. How about if we do it the following Monday then? So, see, Friday would be the 17th, and so Monday I think would be the 20th. Is that right? Okay. Let's plan on the 20th, okay? Now, I am going to still have to make a change to the schedule though. So, the only part of chapter 24 that I'm going to cover is the part on acid-base balance. I think I'm going to skip the rest of it because chapter 23 pretty much explains how sodium in water is regulated. And we're going to talk about potassium today too, if we get that far. So I don't think we need to cover the rest of chapter 24. So we'll just cover the part of chapter 24 on acid-base balance. Okay? All right, so on Wednesday, I talked about the reabsorption of water and electrolytes like sodium and nutrients like glucose and amino acids by the proximal convoluted tubule. Yes?
Are you going to change the study guide then? Or keep it the same?
I'll try to, yeah.
Thank you.
I'll try to change that.
Say thank you.
Okay. I'll take the parts off that we aren't not going to be covered on the test. Okay, so
I said thank you.
today what we're going to do is we're going to look at how about another 20% of the water that's filtered is reabsorbed by the nephron loop, and then how our water balance is fine-tuned by regulating any further reabsorption in the distal convolute tubule and the collecting ducts. Okay? So there's really three different parts of the nephron where water is reabsorbed. Most of it's reabsorbed in the proximal convoluted tubule. They say it's about 65%, but it probably varies a little bit. And then another 20% is reabsorbed in the nephron loop. That goes down into the medulla. And then, depending on whether a person is overhydrated or underhydrated, you may or may not get more reabsorption in the distal convoluted tubule and the collecting ducts. So we're going to look at that additional reabsorption that can take place in those two other parts of the nephron. So the nephron loop reabsorbs another 25% of sodium, potassium, and chloride, and about 15 to 20% of the water in the glomerular filtrate, depending on the source that you get your information from. So the ability of the nephron loop to reabsorb those electrolytes in water is due to an osmotic gradient in the interstitial fluid of the medulla. And that concentration gradient that becomes more and more concentration as you go deeper into the medulla is formed by a mechanism called the countercurrent multiplier. Okay, so that's one of the things we're going to be looking at today. And that mechanism is due to differences in the permeability of the ascending and descending limbs of the nephron loop to water electrolytes. So let's take a look at a diagram of the nephron. So the nephron loop is this part right here that dips way down into the medulla. So this is the cortex up here, and all of this down here is in the medulla, all right? So there's two main parts to the nephron loop. The part where the filtrate first goes down deeper in the medulla, that's called the descending limb, because the filtrate is descending deeper into the medulla. Then the nephron loop takes a hairpin turn, and it goes back up towards the cortex. This is called the ascending limb. The ascending limb is further divided into two parts. There's a thick ascending limb where active transport of sodium, potassium, and chloride takes place. And then there's a thin ascending limb. The thin ascending limb is permeable to electrolytes, but not to water. Okay? So, this part of the nephron loop, the ascending limb, is permeable to electrolytes, but not to water. The descending limb, which is where the filtrate goes through first, is permeable to water, but not to electrolytes. Okay, so those differences in permeability is what produces this countercurrent multiplier, and that's what produces a concentration gradient of electrolytes in the medulla. So this concentration gradient goes from about 300 milliosmoles per liter in the cortex, and the electrolyte concentration increases almost about fourfold by the time you get down into the deeper parts of the medulla. Okay, so to see how this works, we need to start first with what happens in the ascending limb. That's not where the filtrate goes through first, but to really understand how this works, we need to start there. So, by the time the filtrate gets down to this hairpin turn, it has been concentrated because you've already had a lot of reabsorption of additional water in the descending limb. And so by the time you get down here to the deepest part of the medulla, the filtrate, it has an osmolarity of about 1,200 milliosmoles per liter. So it's about 4 times more concentrated. And that's because of this concentration gradient, okay? This concentration gradient causes water to be reabsorbed in the descending limb. And as more and more water gets reabsorbed, the filtrate becomes more and more concentrated. So now we have a very concentrated filtrate. It's going to start moving up. But as it starts moving up towards the cortex, the surrounding interstitial fluid is becoming more and more dilute. So as this filtrate moves up through the thin ascending limb, as it's moving up, the surrounding environment is becoming more and more dilute. And so when that surrounding environment becomes more dilute than the filtrate, then electrolytes like sodium and chloride and even potassium are going to diffuse out into the surrounding interstitial fluid. That adds to this concentration gradient here and helps maintain that concentration gradient. Then as the filtrate gets up here into the thick ascending limb, there are their primary active transport proteins that co-transport sodium, chloride, and potassium all at the same time. This is active transport, so it requires energy in the form of ATP. So that there are these active co-transport proteins that simultaneously actively transport sodium, chloride, and potassium into the interstitial fluid surrounding these tubules. That active transport is what initially establishes this concentration gradient. And then the diffusion of sodium and chloride out of the thin ascending limb, that's what maintains that higher concentration gradient in the deeper parts of the medulla. Okay? So, now that we have this concentration gradient, when the filtrate starts moving down the descending limb is permeable to water, but not to electrolytes. So you're starting out with an osmolarity of 300 milliosmoles in the proximal convoluted tubule. When it goes down into the descending limb, the interstitial fluid is more concentrated. So that creates an osmotic gradient. That osmotic gradient then is going to draw water out of the descending limb, making the filtrate more and more concentrated. Because it's concentrated, when it moves up, then sodium chloride is going to move out. So the ascending limb produces this concentration gradient. The descending limb then reabsorbs about another 15 to 20% of the water that's filtered. Okay? So that by the time that the filtrate gets back up to the distal convoluted tubule, we've reabsorbed a total of about 80% of the water that's filtered. But that leaves another 20% that is still remaining in the filtrate, in the distal convoluted tubule. And if we lost all of that 20% of fluid, that would amount to a significant fluid loss. And so there's additional reabsorption that can occur in the distal convoluted tubule and also in the collecting ducts. But that additional reabsorption that can occur in these two places depends on the osmolarity of our body fluids. Okay? If we are very dehydrated and the osmolarity of our body fluids is high, then more water will get reabsorbed in the distal convoluted tubule and the collecting duct. But if we're overhydrated because we've been drinking lots and lots of water, then not very much water will be reabsorbed at all. And so whatever's not reabsorbed is going to be excreted in our urine. Okay, so we'll produce larger volumes of urine to bring the osmolarity of our body fluids back to normal. Okay? So next thing we need to look at is how we can get hormonally regulated reabsorption of water in the distal convoluted tubule as well as the collecting ducts. And there's two main hormones that are involved in that hormonally regulated reabsorption. The first one is aldosterone. Aldosterone regulates reabsorption in the distal convoluted tubule. And the other hormone is ADH. ADH regulates further reabsorption of water from the collecting ducts. Okay? All right, any questions? Okay, so this countercurrent multiplier, the way it really works is it works by a positive feedback mechanism. And this diagram is from your textbook, and it just describes this positive feedback mechanism. So the first step is that you're always getting water and electrolytes like sodium and chloride. You're always getting that filtered into the proximal convoluted tubule. So you're always getting more salt added to the proximal convoluted tubule. Much of that gets reabsorbed though, but there's still quite a bit that remains when that starts going down to to the nephron loop. So as this filtrate moves down, the higher the osmolarity of the extracellular fluid, the more water leaves the descending limb. The more water that leaves the descending limb, the more concentrated the filtrate becomes inside the lumen of the tubule. So by the time you get down here to this hairpin turn, it's been concentrated about 4 times. So then as that concentrated filtrate moves up, the surrounding interstitial fluid becomes less and less concentrated. So that produces a gradient that allows for passive diffusion of sodium and chloride and potassium out the thin ascending limb. And then when you get up to the thick ascending limb, more electrolytes are actively reabsorbed using sodium potassium chloride symporters or co-transporters. And those are actively, and so both the diffusion of electrolytes out the thin ascending limb and the active transport over the thick ascending limb, both of those are what produce that concentration gradient in the medulla. So the more salt that's pumped out of the ascending limb, the saltier the extracellular fluid is in the renal medulla, and the more water reabsorption you can get. Okay. So one of the factors that affects how much water you can reabsorb in that nephron loop is actually the length of the nephron loop. Because the longer that nephron loop is, the more concentrated the extracellular fluid becomes. And I learned about that little fact because when I was in graduate school, one of the fellow graduate students had done his master's thesis on the length of the nephron loops in animals that lived in deserts. And if you look at animals that live in deserts who don't have to drink much water at all, they get almost all their water just from the food they eat. That's because they can really form a really super concentrated urine. They have really long nephron loops. If you look at animals that live in very moist environments, like near streams or in a rainforest, they tend to have a lot shorter nephron loops because if there's lots of water in the environment, they can find plenty of water to drink. They don't need to form as concentrated urine, so they tend to have shorter nephron loops. So there are some nephrons in humans that are called cortical nephrons, where they're located closer to the cortex. And then there are other ones that are located that are called medullary nephrons, where the nephron loops dip way down into the medulla. It's the medullary nephrons that are able to reabsorb more of the water, more so than the cortical nephrons. Okay, so by the time the fluid arrives at the distal convoluted tubule, about 20% of the water and about 7% of the salts are still remaining. And if none of that was reabsorbed, we would lose about 36 liters of water a day. That's a lot of water. 36 liters is what? There's a little over 4 liters to a gallon. So that's about, 9 gallons of water a day we'd be losing? That'd be a lot of water to have to replace. You'd have to drink a lot of water. So the distal convoluted tubule and the collecting ducts reabsorb additional water and salts, but only if necessary. In other words, the distal convoluted tubule and collecting duct is where fine-tuning of fluid and electrolyte reabsorption occurs. Okay. So I thought of a good analogy of this to help you remember the relative amounts of water that are reabsorbed in the different parts of the nephron. But I think I'll wait until we go through reabsorption in the distal convoluted tubule and the collecting ducts before I show you this analogy that I just came up with off the top of my head earlier today. So, One of the hormones that regulates reabsorption of water in the distal convoluted tubule and the proximal collecting ducts is aldosterone. And aldosterone, if you remember, is secreted in response to angiotensin II production, which is produced as a result of that renin-angiotensin-aldosterone system. So if blood pressure drops, if the blood pressure to the afferent arterial drops, then the juxtaglomerular cells or granular cells secrete renin. Renin breaks off a part of a plasma protein called angiotensinogen, form angiotensin 1. Angiotensin 1 is then converted to angiotensin 2 when another part of it's broken off. Angiotensin 2 then stimulates aldosterone secretion. Okay? So, if blood pressure drops, then that will stimulate aldosterone secretion via the renin-angiotensin-aldosterone system. Okay? But drops in sodium, as well as increases in potassium, can directly stimulate the release of aldosterone from the adrenal glands, the adrenal cortex. And so what that does, aldosterone increases the number of sodium-potassium ATPase pumps in the basolateral membrane of the cells that make up the walls of the thick ascending limb, the distal convoluted tubule, mostly the proximal portion of the distal convoluted tubule, and the cortical collecting ducts. That results in increased sodium reabsorption, and when you reabsorb sodium, chloride follows the sodium because chloride has an opposite charge, and opposite charges attract. And when you reabsorb more sodium, that increases the osmotic gradient, and so you automatically reabsorb more water, because water always follows sodium. So additional fluid and water is retained, and additional potassium secreted. That way, if this was caused by a drop in blood pressure, that increased blood volume will bring blood pressure back up. If it's in response to a decrease in sodium, that increased sodium reabsorption is going to bring sodium levels back up. And potassium is always transported in the direction that's opposite sodium. So if this aldosterone secretion was in response to elevated potassium levels, this increase in the activity of the sodium-potassium pumps is going to cause potassium secretion, because potassium is always transported in the opposite direction. And so that potassium will be secreted, it will be excreted in urine, and that will bring potassium levels back down to normal. So ordinarily, the direction that sodium and potassium are transported is just the opposite direction. So whenever sodium reabsorption increases, potassium is secreted. Okay. So if blood pressure goes up and sodium levels increase or potassium levels go down, then just the opposite would happen. Less renin would be secreted. With less renin, you get less angiotensin 1, less angiotensin 2. With less angiotensin 2, you have less aldosterone secretion, and so you won't get as much sodium reabsorbed. And less water will be reabsorbed. Okay. And so that will result, if you reabsorb less water, then more water gets excreted in the urine, and that will bring your blood volume back down, and that will help to lower blood pressure. Okay. So basically, this system, this renin-angiotensin-aldosterone system, it just restores homeostasis. That's what it does. So if blood pressure gets too high, response is to bring blood pressure back down again by excreting more water. If blood pressure gets too low, then more water is reabsorbed, blood volume increases, and that raises blood pressure back down again. So it does just what you expect. It's very, very predictable. Okay, now, in addition to aldosterone, there's another pair of hormones that are involved in regulation of sodium. And these two hormones are actually two of the most recent hormones that were discovered that affect the kidneys. They're called natriuretic peptides. Natriuretic peptides can increase sodium secretion and therefore excretion independently of the renin-angiotensin-aldosterone system. And there are two different natriuretic peptides that are secreted by the heart. So these are hormones that are actually secreted by the heart. And they're both secreted in response to stretch of the heart. So as blood volume increases, which can cause increased blood pressure, more blood returns to the heart, and that's going to cause the chamber of the heart to stretch. And when the chambers of the heart stretches, that causes the heart to release these two natriuretic hormones, or natriuretic peptides. The first one is called an atrial natriuretic peptide, or atrial natriuretic hormone. That's secreted in response to atrial stretch. And that happens when blood volume increases, which results in increase in blood pressure. And atrial natriuretic peptide then goes into the bloodstream, and eventually it makes its way to the kidneys, and it decreases reabsorption of sodium. So it has the opposite effect of aldosterone. Okay. And if you decrease reabsorption of sodium, then you end up excreting more sodium. And if you excrete more sodium, you end up losing more water, because water always follows sodium. Okay. That's going to bring blood volume back down, and that's going to bring blood pressure back down again. So it's going to maintain homeostasis. There's also another natriuretic peptide that originally was first called brain natriuretic peptide because it was first isolated from brain tissue. But it's not produced by the brain. It's actually produced by the ventricles of the heart. And so now it's more commonly called B-type natriuretic peptide, or B-type natriuretic hormone. That's secreted in response to ventricular stretching. So if your blood volume increases, which causes your blood pressure to increase, you're going to have more venous return to the heart. The more venous return you have, the more blood is going to fill the ventricles, the more the ventricles are stretched. So if that happens, then the ventricles release B-type natriuretic peptide, and again, that decreases sodium reabsorption, which is going to increase sodium. Whatever's not reabsorbed ends up being excreted in the urine. Okay, so that increases sodium excretion. Okay, so both of those hormones increase excretion of salt, the water in the urine, thereby opposing the action of aldosterone. Okay, so we kind of have two different mechanisms involved in regulation of sodium and water, because sodium and water are their reabsorption are tied together. Their reabsorption or excretion are tied together. Okay. Now, the word natriuretic peptide, natriuretic, comes from the word natrium. And natrium is the Latin word for sodium. So that's where the word natriuretic peptide comes from. So I actually first heard about these hormones when I was in graduate school, and I took an endocrinology class, which was a long time ago. And that was about the time that these hormones were first being discovered. So it was brand new. In fact, it was presented to us as that, these are hypothetical hormones that, it was still a hypothesis that these hormones existed. Now it's pretty much been confirmed And another interesting thing is that B-type natriuretic peptide is sometimes used therapeutically. It's artificially administered to people who have congestive heart disease because it increases sodium excretion. When sodium is excreted, water is excreted, and that reduces blood volume. And congestive heart failure is caused when the heart cannot pump all the blood it receives. And so it's the heart, blood starts accumulating in the ventricles if it can't pump out all the blood it receives. And that causes the ventricles to stretch. As the ventricles are stretched, they become even more weaker, so they pump out less blood they pump out. The more they stretch, the weaker it gets. It's kind of a vicious cycle until the heart just completely fails to function, and the person dies. So one of the ways that you can treat congestive heart failure is to give the person a diuretic that causes them to excrete more water. That reduces blood volume. It brings the heart back down to closer to its normal size. So it disrupts that vicious cycle. Well, B-type natriuretic peptide is one of the drugs that can do that because it increases sodium excretion along with water. All right. Then finally, the last hormone that affects water reabsorption is antidiuretic hormone, or ADH. ADH is also sometimes known as vasopressin. And ADH is actually secreted secreted at the posterior pituitary. It's actually produced in the hypothalamus, and then it's transported down to the posterior pituitary, where it's released. So it's really a hypothalamic hormone rather than a hormone of the posterior pituitary. But we still call it a posterior pituitary hormone because that's where it's released from. So ADH is secreted in response to dehydration, which results in increasing blood osmolarity. And what triggers ADH release is that there are osmoreceptors located in the hypothalamus. So when the osmolarity of the blood increases, that stimulates these osmoreceptors that triggers ADH release from the posterior pituitary. What ADH does is ADH causes the incorporation of additional water channels called aquaporins in the collecting ducts, which makes the collecting ducts more permeable to water. And the collecting ducts go down through the medulla of the kidney. And so as the filtrate goes down through the collecting ducts, the surrounding environment is more and more concentrated. So that draws water out of the collecting ducts. But that only happens if ADH is present, because ADH is what makes those collecting ducts permeable to water. If you don't have ADH, then you don't get any additional water reabsorption in the collecting ducts. So that water just goes out with the urine. So you can get additional reabsorption of water as urine flows to the collecting ducts because it's experiencing a higher and higher surrounding osmolarity as it travels downward through the medulla. So by the time the filtrate goes all the way through the nephrons, you've reabsorbed 85 or 90% of all the water, all the useful electrolytes, all the nutrients, everything that the body needs, and what's left over is a fairly concentrated waste. Okay? And that concentrated waste has a high concentration of urea. And so, actually, surprisingly, in addition to additional water reabsorption in the collecting duct, something else that's reabsorbed is urea. Whoops, let's go back. Got to go back to this diagram of our. Okay, so this is the collecting duct. So in the presence of ADH, water channels called aquaporins are incorporated into the collecting ducts, and that allows more water to be reabsorbed. At the same time, surprisingly, about half of the urea that's in the filtrate ends up getting reabsorbed. Now, to me, that probably sounds surprising because urea is a waste. It's a waste product. That's why we have kitties, to get rid of that urea. But half of it gets reabsorbed. And the reason that it gets reabsorbed is the additional urea that gets reabsorbed contributes to this concentration gradient in the medulla of the kidney. So there's two different things that contribute to this concentration gradient. One is the reabsorption of electrolytes in the nephron loop, and the other is the reabsorption of about half of the urea that flows through the collecting ducts. And so if you reabsorb half of the urea, that means the other half is still going to remain in the collecting duct, and it's going to get excreted in the urine. And so even though we're not excreting all of the urea that's filtered, a lot of it gets recycled. Actually, it goes into the interstitial fluid, and then a lot of that gets picked up by the nephron loop, and then it just cycles back and forth. And that helps to contribute to this concentration gradient in the medulla. But the half that doesn't get reabsorbed, it gets excreted, and by excreting half of all of the urea, that's enough to reduce the blood levels of urea to safe levels. Okay, so you don't have to get rid of all of it to bring your blood levels of urea back down to normal. Okay, so when these mechanisms of urine processing were first discovered, they thought it was just these electrolytes that contribute to this concentration gradient. Then they discovered that actually there's a lot of urea in the interstitial fluid that gets reabsorbed from the collecting ducts. That also helps to contribute to that concentration gradient. So what I want to do then next is I want to show you an analogy that I thought might be helpful to remember where the water gets reabsorbed in the nephron. Okay, and I've heard of other analogies before. I like my idea better. that's just me. I'm probably biased though. So the analogy I thought of would be, and it's probably something a lot of you aren't familiar with. And actually, this predates me too, so I'm not this old. It's called a wringer washing machine. It's what they used to use to wash clothes, probably like when my mom and dad were growing up, because these are pretty old. It was before they had automatic washing machines that run off electricity. Some of these actually were run with a hand crank. You had to crank it to get it to work. But anyway, I found a little YouTube video that shows how this works. So I'm going to show you how this works, and I'll explain how we can use this as an analogy for water reabsorption in the nephron.
Families scrubbed clothes by hands on washboards, wringer washers. They made the job way easier. You'd wash your clothes in a big tub of soapy water, then feed them through 2 rollers. However, wringers were also known for accidents. Fingers and even long hair would often get caught between the rollers. Despite that, these machines revolutionized laundry until automatic washers took over. Did you ever try doing laundry with one of these?
Probably not. So I don't think they were around very long because, like they said, people get their hair caught in it and rip their hair out of their scalp and get their fingers in there and get their fingers crushed. So it didn't take too long before they came up with a better idea for how to wash your clothes. So this is the way I think that this is an analogy for water reabsorption in the nephron. So you first put your clothes, your dirty clothes, in that tub of water. Okay, and there's an agitator in there, and it washes your clothes. When that agitator's done, and some of them you had to crank to agitate it, then you'd reach into the tub and you'd pull out your clothes. Okay. When you pull out your clothes, most of the water remains in that tub. So that's like the water that gets reabsorbed in the proximal convoluted tubule. Okay, that's the water in the tub. Then you take the clothes and you run it through the wringer. That's those two rollers. That compresses the clothes and that takes even more water out. That's like the water that gets reabsorbed in the nephron loop. Okay? But even after your clothes go through the wringer a couple of times, there's still going to be some water. The clothes are still going to be damp. It's not going to be dry yet. Okay? So what do you think they did then to get the clothes completely dry
Hung them outside.
before they had electric dryers?
Hung them outside.
They put them out, they hung them up outside on a clothesline. Okay? When I bought the house I live in right now, when I first moved in there was a clothesline in the backyard. So probably somebody was still using it back then. So to get the rest of the water out, you hang it up on a clothesline. Okay? And the amount of time you have to hang that up depends upon the humidity of the air. So if it's really humid, you're going to have to leave that hung up on the clothesline for a long time. So that would be like reabsorbing water if you were overhydrated. If you had too much body water, you have to reabsorb more water in the distal tubules. If the air is really dry, you can put it out in the clothesline, and you don't have to leave it hanging up very long. That would be like if you're underhydrated. If you're underhydrated, you don't have to you have to reabsorb more water then. Okay. So hanging up on a clothesline, that's where the regulated reabsorption takes place in the distal tubules. Okay, does that make sense? So another analogy I've seen, and I couldn't find it. I think I saw it in a video clip that was supplied by a different textbook publisher at one time. Was like filling up a volumetric flask with water. On a volumetric flask, there's a line, and you fill up that line. That gives you an exact volume of water, like 1 liter. And so if you're filling up a volumetric flask, you first fill up the bottom part by just putting it underneath a faucet and filling up most of the way. And then you might take a smaller graduated cylinder and carefully pour the water to get it up just before the line. And then to get it exactly to the line, so you have exactly 1 liter water, you add water with a dropper. So the dropper would be like the regulated reabsorption in the distal tubules. Filling up the faucet, that would be like the reabsorption that takes place in the proximal convoluted tubule. So anyway, I thought those are some analogies to kind of help you understand how the nephron works. Okay.
This old machine.
Okay. So, let's skip over parathyroid hormone. That's just a hormone that regulates calcium. That doesn't really have to do much with, it's secreted by the kidneys, but doesn't really have much to do with water and sodium regulation. So this just summarizes the processing of urine that occurs in the nephron. And so in the proximal convoluted tubule, we get reabsorption of glucose, amino acids, smaller peptides, vitamins, lactate, urea, uric acid, and lots of electrolytes. Also, you get secretion of urea, uric acid, creatinine, hydrogen, ammonia, some drugs into the proximal convoluted tubule. Those are things that didn't get filtered, that remained in the blood. Those can be secreted in the filtrate. Then in the nephron loop, you get further reabsorption of water and reabsorption of electrolytes. And then in the distal convoluted tubule and in the collecting ducts, you may or may not get more reabsorption of water depending upon the hydration status of the person. And that reabsorption occurs by way of hormonal regulation. It's hormonally regulated. So if you're dehydrated, more sodium and more water will get reabsorbed. If you're overhydrated because you've been drinking too much water, then less ADH will be released and less aldosterone will be secreted. You'll get less reabsorption of water and you'll get more water excreted in your urine. Okay. Well, we're about out of time, so this is probably, we went over this in lab already, so I don't think we need to go over this again. There are a couple more slides, though, I do want to show you. We can do that on